All right. Hello, everyone, and welcome back. I hope everybody had a good little break. Everybody was able to get their coffee or tea. So just to introduce myself real quick, my name is Jordan McBreen, and I'm a PhD student here in the agronomy department, as well as this year's 3-minute Flash Talk competition chair. So just a bit of a background on the 3-minute Flash Talk competition. The idea is that an effective scientist should really be able to deliver a summarized version of their research in only 3 minutes or less, and that being able to successfully do so really indicates a good scientific communicator, which is key in bridging the gap between research results and their real-world applications, be it in the field or in the consumer marketplace. Effective scientific communication also allows us to productively collaborate with one another by increasing our understanding of each other's works. So let's briefly go over the rules, and then we'll jump right into the presentations. There will be 3 judges who will be impartially scoring each of the presentations. The presentations are limited to 3 minutes maximum, and competitors exceeding these 3 minutes will be penalized with, and I'll be keeping the official time. Time begins when the competitor begins speaking. A single static PowerPoint is permitted. All questions will be saved for a separate time slot for all presenters to respond. And once all the presenters have gone ahead and presented, we'll go ahead and do the Q&A. So if anybody has any questions, be sure to put those in the Q&A as you're thinking of them. And then finally, there will be a 3rd, 2nd, and 1st place winner, with there being cash prizes of $100, $150, and $200 respectively. So be sure to do your best, guys. All right, and so to start off the competition, we have Andrew Biswas. Let me go ahead and I will share my screen, and then Andrew, you can go ahead and take it away. All right, I'll go ahead and I'll start the timer whenever you start talking, Andrew.
Hi, Jordan. Can you hear me?
Loud and clear.
So you want me to share my screen, or you are sharing the slide? I will go ahead with there.
So I'll go ahead and I will share all the slides.
Okay, sounds
Are the
perfect.
slides up there? Everybody's able to see?
Jordan, you're in presenter mode.
Yeah, it's like little icon.
Oh, okay.
Yeah, I think it's better now, but it's not slide mode, I guess.
Go ahead and start it. Start the slideshow. And then
How about that?
there we go.
Now you have to go to my presentation,
right,
I guess.
okay. All right, okay. Go ahead whenever you're ready, Andrew.
Good morning, everyone. I'm Anju Biswas, a PhD candidate from Agronomy Department at University of Florida. I'm excited to share my research entitled High Throughput Approach for Predicting Alfalfa Yield. Predicting alfalfa yield depends on phenotyping. Phenotyping is very crucial in plant breeding. So what you can see from figure 1A, manual phenotyping, which is time and labor intensive. And you can see even my PI also in blue t-shirt helped us there. On the other hand, I am the only person in figure 1B accomplished the same task within 15 minutes. Isn't it cool? So high throughput phenotyping can be an alternative of manual phenotyping. So what's our research objectives? One of our objectives was to phenotype the alfalfa breeding population for harvest accumulation using both manual and using unmanned aerial vehicle. The second objective was to find out the correlation and prediction efficacy of high throughput phenotyping with manually collected harvest accumulation. Our research was conducted in Sitra, 30 minutes away from Gainesville, and experimental design was row column design. We had 145 full sieve and 36 half sieve families, including 3 controls. 4 manual harvests had been done in different time points in 2018 and 2019 based upon 10% flowering of controls. An unmanned aerial vehicle equipped with multispectral camera was flown before each harvest. Then the image processing was done in EasySoft, which is visible from figure 2. We stitch the image using AgSoft and QGIS to generate 4 vegetation indices based on 2 or more bands designed to enhance the contribution of vegetation properties. And figure 2 represents the workflow had been done in that software. Pearson correlation and linear regression was done in R later on. In result section, I'm presenting the result from one harvest as we found similar trends for all 4 harvests. You can see from figure 3, correlation between vegetation indices and harvest accumulation. The x-axis represents 4 vegetation indices and y-axis represents the harvest accumulation. And we found higher correlation, which is more than 80%, and medium prediction efficacy, which is visible from figure 4, which is more than 65% accurate between harvest accumulation and vegetation indices, which you can see already aside figure 4, where x-axis represents harvest accumulation and y-axis represents vegetation indices. So what's our take-home message from this research? High-throughput phenotyping can be an alternative of manual phenotyping to speed up the selection process of alfalfa breeding population, and we will be benefited from that in our breeding program. Thank you, everybody. Thank you for listening me with patience. I will be happy to take any question. Thank you.
All right, great job, Andrew. So next up, we have Julianne Janori. Julianne, the floor is yours.
All right. How many of you guys have actually taken a walk through a scenic downtown or through your local park and actually paid attention to the ornamental plants around you? Chances are you've overlooked one of the most common choices for beautifying these areas. And that plant, as seen in the image in the top left, is Begonia semperflorens, or the wax begonia. These beautiful ornamentals are typically used to line flower beds that line roads, walkways, or home gardens, but they're actually currently encountering some setbacks. Much of the wax begonia production in the US occurs in Florida, yet the hot summers coupled with intense light are actually taking a toll on the growth, leading to a loss of product and revenue for big growers across the state. If you view the panel on the left, you'll see that I conducted an experiment to first characterize the response of various wax begonia varieties to heat and light stress, and second, to see if there are any morphological characteristics that enhance the tolerance of any of the varieties studied. If so, these morphological markers will allow us to easily make beneficial selections in breeding. Now, when it comes to wax begonia, there are certain morphological changes that are uniform across all varieties studied. The stomatal density consistently increases when plants are exposed to heat and light stress compared to them being grown in the shade. As shown in the circular images on the left, the genus Begonia is unique because some species in the genus have stomatal clusters as opposed to a singular stomatal, which aid in gas exchange. Now, there's no actual change in the stomatal cluster sizes in either of the treatment groups. So now here's where it gets interesting. As I direct your focus to the middle of the slide, one of the traits that does vary depending on the species is the degree of leaf folding. Typically, plants fold their leaves to reduce the intensity of direct sunlight, and that looks to be the case here, but the intensity of that folding varies from variety to variety. When plants are exposed to direct light as compared to shade, there's a significant difference in that folding. Highlighted in yellow, you can actually see that one of the varieties, FB059, folded significantly more than the other 3 tested. The commercial green variety, denoted as CG and highlighted in red, barely folded at all there. Another key morphological trait that could perhaps increase the resistance to light and heat is the cuticle thickness. The story is the same as leaf angle, with FB059 not only having a thicker cuticle, but also increasing its cuticle thickness significantly more than any of the other varieties tested. So this is visible in the table highlighted in yellow and in the chart at the bottom. So now that we've identified morphological characters that vary from variety to variety, the question is, how do we actually know that they enhance heat tolerance or light tolerance or any of these abiotic factors? In addition to the morphological characters, I also wanted to know the physiological response of these varieties. As seen in the chart on the right, the ion leakage of FB059 in the unshaded environment is significantly lower than the other varieties. When looking at fluorescence, a great indicator of the stress level of the plant, we also see that FB059 has a higher FV over FM ratio, signifying that the plants are under less stress. The goal for this information is that it can be applied to making breeding decisions to improve the growth of wax begonia in the industry. Thank you.
All right, thanks so much, Julian. Great job. So next up, we have Kara Gerline. So you can go ahead and start when you're ready.
Hi, can you hear me?
Yep, loud and clear.
Okay, great. Hi, everyone. My name is Gurleen Kaur, and I'm a PhD candidate in CLE Lab here at the University of Florida. So we all like tomatoes with a good flavor, either in salad or a salsa. But as tomatoes have gotten larger, they got more water and have become nearly tasteless, with decades of breeding for producer priority traits such as fruit size, appearance, and disease resistance. In contrast, consumer prioritizes the flavor, aroma, nutrition, and sustainability. So there is a need to bring the lost flavor back in modern tomatoes for the consumers. As seen in Figure 2, tomato flavor includes specific amounts of sugars, acids, and the unique aroma, which is composed of over 400 chemical compounds that makes the flavor a complex and an expensive trait for breeding. When you chew a tomato, the flavor goes to the throat and to the olfactory nerves in your brain, and that's where you taste the flavor. Only sugars and acids will lead to a bland tomato. Most of the current tomato plants have lost the chemical compounds responsible for good tomato flavor. So now we are looking for ways to make big tomatoes more flavorful by combining them with diverse heirlooms, as you can see in the figure. My research focus is to identify and validate the genes responsible for flavor chemical compounds. To achieve this, I have used an integrative approach using multiple tools based on genome, transcriptome, and phenotype for flavor compounds. To put all pieces together for the flavor puzzle, we use a variety of genomic tools to identify consensus significant genes responsible for the synthesis of flavor compounds. The purpose of using multiple approaches is to better understand molecular breeding tools related to flavor. I have identified genes associated with flavor compounds, and currently I'm working on gene editing to validate the gene function. So the overall implication of my research work is to unravel significant genes critical to improve tomato flavor, and that could be a step closer to make big tomatoes taste better for the sake of our consumers. Thank you very much for listening.
All right, great job. Thank you so much. So next up, we have Lise Porto.
Yes.
So go ahead whenever you're ready.
All right. So hello, everyone. I'm Liz Porto. And about 2 years ago, I've moved to Florida to pursue my master's. And since then, I've noticed the importance of having an umbrella because Florida's climate conditions is unique, presenting warm temperatures and high humidity. And these conditions are very attractive for disease development. And lettuce crop has many diseases affecting its production. However, the foremost important disease of lettuce is downy mildew, caused by Brimia lactuca. And this pathogen is very genetic diverse, presenting several races. And the race structure is defined by using a set of differential cultivars with known resistance or susceptible reaction to this pathogen. So the idea of this work was to use the same lettuce downy mildew differential cultivars to be tested with other lettuce diseases in order to find common resistance in the lettuce crop. So 3 different isolates of Xanthomonas hortorum petovarvitens, the pathogen of bacterial leaf spot, were tested, were inoculated in the 17 differential cultivars. And after 10 days, these plants were evaluated for disease severity using a rating scale varying 0 to 5. Non-parametric analyses were conducted based on the relative marginal effects. 2 cultivars, Muriatis and Sylvanas, had low disease severity similar to the resistant control. The same idea was applied to Fusarium wilt experiment, where the roots of the differential cultivars were merged into a suspension with Fusarium isolate, and weekly, these plants were assessed for root discoloration and foliar damage using a rating scale varying 0 to 5. And the relative marginal effects showed that 3 differential cultivars Silvinas, Ballesta, and Colorado presented even lower disease severity than the resistant control for both parameters analyzed. So my research suggests that identifying multiple disease resistance in a single accession is possible. And in a scenario of climate change, where resistant outbreaks are suspected to be more frequent, the identification of accessions with multiple resistance is key for breeding programs, since, unlike myself, lettuce plants cannot use umbrellas. Thank you.
All right, thank you so much, Lise. So next up, we have Lushan Gamir. Lushan, whenever you're ready.
For some reason, I'm not able to on my video.
Oh, that's okay. No worries.
I think it's good now. Okay. Hello, everyone. My name is Lushan Gimire, and I'm currently a PhD student on a PMCB program working with Dr. Patricia Munoz in Blueberry Breeding and Genomics Lab. So now moving towards my presentation, plant breeding programs, they need to be aware of current and emerging threats to ensure future food security, especially for perennial crop plants such as blueberries, for which breeding is time intensive and resource intensive. It is very critical to identify the regions in genome that are associated with desirable traits if we really want to accelerate the breeding process. Annual Florida Blueberry Growers Surveys for breeding trait priorities has ranked fungal disease and anthracnose among the most important disease in terms of incidence. Not just decrease in production due to this disease, it has already decimated planting of commercial cultivar flicker, which is a common parent in many hybrids. Therefore, to deal with this issue, we aim to identify genomic regions associated with resistance against this disease. Now for this, a multi-parental population in a 4x4 half diallele design was built with a total of 700 individuals. And I'll briefly tell you about how a 4x4 half diallele design looks like. It is exactly, the design looks visually exactly how it is shown in the gray-white table here in the middle with 4 different parents. And one thing to note, all these parents exhibited different levels of resistance and susceptibility in the field. Now, for phenotyping post-pathogen inoculation, every individual was given a score by observing its disease severity as shown in the photo with leaves here below the table. And the same population was genotyped using capture sequencing. Now, this method of sequencing allows us to extract genomic information from the regions of our interest. Now, let's look at this graph to our bottom right here. Preliminary results from phenotyping showed that the crosses were segregating for the percentage of susceptible plants. Additionally, further analysis showed that some crosses were working better or worse than would be expected from them, which means that specific combining ability of each parent was significantly different. This has hinted us towards potential polygenic genetic architecture of this trait. Now, the next step will be to link phenotypic and genotypic data to identify those associated genomic regions. This way, we can understand the genetic architecture of this trait, accelerate the development of resistant cultivars, and minimize losses due to disease in blueberry fields. Interestingly, this pathogen has a broad host range, so resistant genomic regions found in blueberries also have the potential to be introduced into other host species as well. Thank you everyone for listening.
All right, thank you so much. So moving right along, next up we have Mithri Bikasani. Mithri, whenever you're ready, you can go ahead and start.
Yeah, I would like to start now and wish you all a great day ahead. And I'm Bikasani Maitri from Indian Council of Agricultural Research, Indian Institute of Millets Research, located in southern part of India in Telangana state, Hyderabad. And this ICAR IIMR is one of the previous agricultural public research institutes in India, dealing with basic and strategic research exclusively on sorghum and other millets like pearl millet, proso millet, finger millet, foxtail millet, barnyard millet, kodo millet, and together they count for about 8 millets. So yeah, coming to my crop, that is proso millet, which is Panicum miliaceum, also called proso millet, broom corn millet, or even white millet in France. So this is domesticated in China years ago. And the crop has specific notable features like rich amount of protein among all the cultivated cereals, which is almost like 13 to 17% over wheat or rice. And then the crop has, the grains has almost very good energy, and the crop is also gaining importance as a biofuel crop, and the studies are yet to be out. And this crop especially has highest water use efficiency, that is highest harvestable yield per water use among all the cultivated cereals. So yeah, like until last year, the grass, which is Panicum virgatum, which is the closest relative to this crop. So till then we thought of using only the switchgrass genome sequence to filter out the SSR markers. So these markers are now mined out using the proso millet genome sequence, which is released only a year ago, and using the GMATA, that is genome-wide microsatellite analyzing toward application, wherein the sequence from NCBI is taken and inserted in this bioinformatic tool, and through which we can really filter out the desired SSR markers by applying various filters like the number of repeat motifs and number of base pair lengths, and finally we have come out with many SSRs, but out of which we have to really reduce it to 90 SSR markers based on various parameters like number of AT repeats and GC content among the markers that were out, and we have to really eliminate out primers for those loci which are very critical to design any primer, and only those with A and T and those for which the two loci are very close. So keeping in parameters of all these together, we have filtered out 90 SSR markers, and we are really screening out them using 24 genotypes to check for their polymorphism, and yes, they're really working well. As you could see, the gel image of one of the markers with which we have run PCR using 24 genotypes. And ultimately, these are the first stage SSR markers that are out from the main reference genome of proso millet. And I hope these markers would definitely help in studying the genetic diversity studies of proso millet that are located worldwide. Thank you.
All right. Thanks so much, Mitri. Great job.
Yeah. Thank you.
So next up, we have Pablo Sipowicz. Pablo, if you're in here, if you can go ahead, then the floor is all yours.
Hi, can you hear me?
Yep, sounds good, Pablo.
Okay, perfect. Hello, my name is Pablo Sipowicz. I'm a student, I'm a PhD student at Dr. Rios lab. I will present our studies in genetic diversity in wild and domesticated annual ryegrass. Annual ryegrass is the common name of the species Lodium multiflorum. It's mainly used as a forage crop, and it's self-incompatible. So when we describe accessions in annual ryegrass, we should think about highly heterogeneous populations. The University of Florida's ryegrass breeding program has successfully exploited this heterogeneity by releasing more than 20 cultivars in the last 20 years. However, in the recent years, there has been evidence of or signs of germplasm exhaust. And to tackle this issue, the breeding program has selected 139 accessions from the USDA's National Plant Germplasm System to try and broaden the genetic diversity. These accessions can be divided by their passport status in wild type, cultivated, and unknown. So the objectives of our studies were to assess the genetic diversity in the USDA's green collection, the University of Florida's breeding program, and some commercial cultivars. And for this, we genotyped the materials with single sequence repeat markers. In the new collection, 50 experimental lines from our breeding program and 13 commercial cultivars. And the data analysis indicates that there's a presence of population structure in the collection that we analyzed. And this is important because if the collection is going to be used for association studies to discover QDLs of interest, there should be a correction for the population structure to avoid bias. And data also suggests that the wild type accessions that were selected are genetically distant from the rest of the materials that were analyzed, indicating that the addition of the wild type accessions will definitely broaden the genetic diversity of the germplasm. And our future interest of research will be centered in developing a genomic selection scheme to integrate traits of interest in our breeding program. Thank you very much.
All right, great job. So moving right along, next up we have Kasim Ali. Kasim, whenever you're ready.
Can you hear me?
Yep, loud and clear.
Hello everyone, my name is Kasim and I'm a researcher in Dr. Peter's lab in agronomy department here at UF. Biodiesel is a renewable and environment-friendly source of energy, mostly derived from oilseed crops like canola and soybean. Metabolic engineering of high biomass crops like energy cane for the production of oil is a promising strategy to improve the oil yield of traditional oilseed crops. Energy cane can be converted into an oil cane by co-expression of 3 lipid genes through a complex metabolic engineering approach. Figure 1 shows a typical engineered metabolic pathway for hypercumulation of lipids. In this, the first 2 steps is to increase the fatty acid synthesis and triacylglycerol tag molecule, which is the high storage lipid molecule. Third one is to optimize its storage and finally preventing that tag from hydrolysis. So involvement of these 3 lipid genes make this metabolic engineering challenging. Comparison at single gene level would be simple but not conclusive because each variant needs to be evaluated in context of at least 2 other lipid genes. Therefore, using constitutive promoters expressing all the genes continuously may require a large number of transgenic lines and samples to be analyzed to find the few that are informative. In this scenario, inducible expression system in which gene of interest is only expressed in the presence of a specific inducer is a promising strategy to accelerate the design, test, build, and learn TPTL cycles. So for this, the objective of our study is to explore if we can accelerate this TPTL cycle for hypercumulation of lipids in energy cane by using a inducible transposition system. Our hypothesis was that using a heat shock inducible promoter for 1 or 2 lipid genes while constitutive promoter for all other genes will facilitate the variant comparison. For this, figure 2 shows we transformed the energy cane calli using a biological gene transfer with lipid genes in which the candidate genes under the control of HSP promoter. Results. Our results showed that energy cane can tolerate the induction of lipid genes with heat shock promoter with multiple cycles of heat treatment at 40 centigrade without necrosis. Figure 3a, this graph shows a time course experiment of heat treatment that upregulates the gene expression of 2 important lipid genes, DGAT and Wrinkle1, from 96-fold to 150-fold. And subsequently, that led to increased oil production from 5-fold to 16-fold in stem and leaf tissues, just within 4 days of gene activation. Conclusion. So this strategy has a great potential to accelerate the TPTL cycle for identification of superior lipogenic factors, variants in energy cane for biodiesel production. Thank you.
All right, thank you so much. Great job. Okay, so finally we have Swati Shrestha. And go ahead whenever you're ready, Swati.
Thank you, Jordan. I'm excited to share my research with you all. Either as vegetables, soup, or pie, squash and pumpkin always find way to our home and heart. However, profitable production of squash and pumpkin in the grower's field is threatened by the infection of a virus named papaya ring spot virus, or PRSV, because popular squash varieties like butterbush are susceptible to the virus. Thus, there's a need to develop PRSV-resistant squash lines in the future breeding program. Luckily, natural source of resistance to PRSV has been reported in a local squash landrace from Nigeria called Nigerian local, and many breeding programs seek to transfer this resistance into commercial lines. Unfortunately, genes and QTL associated with resistance remain unknown, making the breeding program tedious and less effective. Now, many of you here might know what a gene is. Let me tell you what a QTL is. In plants, numerous genes with minor effects control most of the agronomically important traits, and the region of those genes is known as QTL, or quantitative trait loci. Through my research, I have been trying to identify QTL associated with PRSB resistance in squash by using a whole genome sequencing-based bulk segregant analysis technique called QTL-seq. In fact, by using this method, I have already identified QTL associated with PRSB resistance in squash in chromosome 9, as can be seen in the bottom right figure of your screen. After QTL identification, I have also developed molecular markers tightly linked to PRSB resistance from this QTL region. Now, what's the implication of all this? Let me explain this with an example. If we consider the genome of a plant as a university, then various departments can be considered as QTLs controlling different traits. Let's say you want to put up a play by Shakespeare. You would choose students from drama department instead of IT department in order to get the best performance. Likewise, plant breeders and geneticists should now focus on QTLs and markers identified through my research for the rapid selection of PRSB-resistant individuals in each generation, thus accelerating future breeding programs through marker-assisted selection. Thank you.
All right, great job, Swati. So I'll go ahead and I'll stop sharing my screen here. Just want to say thank you to all of our participants. Let's give all of our presenters a virtual round of applause. Thank you so much, guys. You all did a great job, and you each taught us a lot about each one of your areas of research. From here we can open up the session to a few minutes of Q&A. So if you have any questions that you haven't submitted quite yet, please feel free to do so now. So I'm going to open up the Q&A and I'll see what happens. So here's a question for Julian. Do you know the ploidy level of the varieties that you tested, and is there a different response related to ploidy?
Yeah, actually, I love that question because it really kind of follows the path of my entire PhD. So that's actually what I'm working on now. Begonia is a mega diverse genus. It's over 1,900 identified species at this point, and new ones are being released every month. Accompanying that diversity, there's also a huge diversity in ploidy level and in genome size, both of which can have an effect on the abiotic resistance or tolerance, if you will. So have I done it yet for any of the 4 varieties? I've done it, but I haven't been able to analyze it yet. So short answer, I can't tell you. But based off the parents, for example, of FB059, I think right now we believe it to be a tetraploid, but again, not definitive yet on that. So thank you.
All right, thank you. So I've got another question here. This one's for Anju. Anju, can you talk a little bit about the different covariates and challenges you ran into in your HTP data collection with both the UAV and the ground sampling that you guys did?
Hey, Jordan, can you hear me?
Yep, loud and clear.
Can you please repeat the question?
So can you talk a little bit about the different covariates or just general challenges that you ran into when you were collecting UAV-based data, and as well as that ground truth data that you took, and were there any problems in finding correlations there?
Okay, so first of all, flying a drone or unmanned aerial vehicle, it's sometimes challenging. It depends on weather and some other things. Like if it's cloudy, you cannot fly the drone. So we need full sunlight first of all. So it's more about flying part. And for data part, when we get the data, though you see, like, I'm the only person who was flying the drone, like taking the all spaces from other, lab mates or labor from there. The thing is, it takes technical skill to process the data in different software to generate vegetation indices. And when we analyze the data, as I showed you in Pearson correlation, we really found very good result for that. And one of our paper already published, we found that applying high throughput data, we can even replace manual harvest accumulation by like up to 50% or even 70% in some cases. Even still we will get the similar prediction efficacy. So I would say it's really good for our breeding program, especially for perennial alfalfa. You know that it's very tiring and tedious when we collect data each month. Generally, we do each month, and we need like lots of laborers, not only for harvesting, but also for like after processing things, drying, taking weight, and data input. Lots of things are there. Yeah, thank you.
Thank you. So I've got a question here for Swati. Which methods did you use for QTL discovery for the squash resistance?
Oh, thank you for the question, Jordan. So basically, we used a whole genome sequencing based bulk segregated analysis technique. So in that technique, what happens is you collect 10 of the resistant plant samples and 10 of the susceptible plant samples, and then you send those bulk population for sequencing. And after getting the sequencing results, you look at those two bulk samples, genomic region, and then look at the SNPs associated with them and try to locate the SNPs and then the QTL. I saw a question in chat as well. I think that's related to me. So since I'm talking, I will answer that question as well. The question is, do you think QTL still works because we have developed CRISPR-Cas9 technology to modify the gene? So basically, I think for my crop, which is squash, yes, because a lot has been done in other crops like rice and wheat, but for squash, there hasn't been a lot. And for the virus disease and squash that I am working on, we just have like basic inheritance studies. So we still don't know what gene is working on or what QTL is working on for the resistance. So for my crop, yes. But also I would like to add that even for CRISPR and other modern techniques, we need to locate and identify the gene. And the basic step for that is QTL identification. So everything starts with that. And in my case, the QTL identification is like based on whole genome sequencing. So we can locate the SNPs very accurately. And then that's the step. And the next step is we are thinking of maybe using some like genome editing technologies in our lab as well, if we have like available resources in the future. So that's all. Thank you.
All right. Thank you. Good answer. Okay. Here's one I have for Kasim. So it says you showed that you guys use the gene gun for transformations. Does that involve using gold nanoparticles, and how efficient is this transformation method in making successful transgene deliveries?
Thank you. Yeah, we used the gold particle to bombard, and it is quite successful compared to other transformation method like bacterium.
Okay. Yeah. All right. Sounds good. Here's one I've got for Kaur. What are some of the challenges of scientifically improving taste, which can be a very subjective thing in tomato?
Thank you for the question. As simple as a slide looked, it is nowhere close to when we work on flavor. As I said, like it's complex in terms of phenotyping and in terms of money and everything. So with the subjective nature, when we do, so our lab does taste panels for other studies. It's not part of my project yet. So we do taste panels and all, but it's dependent on season. It dependent on like multiple factors that I didn't, explain to just for the sake of simplicity here. So all in all, in a nutshell, it's very complex and there are multiple pathways. So that's why our lab is only focusing on flavor for so many years. And it's a lot has been done, but we are not there yet. So yeah, I don't know if that answered the question.
Yeah, I think so. Definitely. Definitely. Let's see. I think we may have a couple more questions. So here's another one for Julian. It says, so you mentioned that you're taking a kind of a suite of different morphological traits and morphological data. Are you using any high throughput phenotyping methods for that? And if so, could you kind of elucidate those a little bit?
I am not. I'm not doing that at this point because they're really, as far as research goes, there's not too much, not as much money in ornamentals compared to, you know, your fruiting crops or anything like that. So stuff like that does cost quite a bit of money, and begonia isn't quite at the developmental stage where I think we can actually successfully apply it. Yeah, I guess you could say I'm the first in a line of many PhD students who will be working on this project. So I'm setting the foundation, if you will.
That's good. That means that everybody will be citing you first.
Yeah, hopefully.
Yeah. Okay, here's another one. This one's for Lushan. It reads, what other possible species do you think your blueberry research may be expanded to in the future? I know you mentioned that it could be expanded going forward. So what are some of those areas you think that could expand to?
That's a great question. So considering the disease that I'm working on, anthracnose, it is caused by Colletotrichum species. So, so far it has a very broad host range, including tomatoes, strawberries, potatoes, and eggplants. So these are the potential species where we can probably test the identified genomic regions. And there should be some more species that I'm not very that I have explored yet, but again, so far my priority being blueberries. I'm just trying to focus more on that. But yes, I mean, all the species that does have that is affected with anthracnose, with Colletotrichum species, we can possibly test the potential.
Okay, here's a quick question. Do you ever get tired of eating all the blueberries that you guys have in that
Yes,
lab?
I do.
Oh yeah, I'm
Yes,
sure.
I do.
All right, here's a question. This is not pointed to anyone specifically, but I think Swati, this is right up your area, and if anyone else has an answer to it as well. So it's, what are the challenges in identifying QTLs and genes?
I think this question is not directed to only me. I know Gurleen's research and even Lucian's research kind of is similar. Yeah, I can give my perspective about it. So that's a good question. Like, what are the challenges for identifying genes and QTLs? Basically, I think that for QTLs, there are a plethora of methods that can be used for identifying QTLs, and each method has its own pros and cons. I can talk about my research, my method that I have been working on since like last 2 years. So in my method is really quick. What we do is we just have developed F2 population, take 10 resistant and susceptible, send them sequencing, get back the results and just analyze it bioinformatically. So in my case, we get like big QTL regions, like they can be like 1 million base pairs. And from that QTL region, if you have to narrow down and find the genes, that gets really difficult because there are like so many genes in that region, and then to specifically find the gene that is related with my disease resistance. So I think that's probably one of the big challenges. But then again, we have like so many softwares available, some of them really accurate. Like the one that I use is called SNP effect software, which can really pick out the SNPs from those regions that have very high impact. And then that has very high accuracy. So in my case, that's the thing, like big, like narrowing down the genes from those big regions is kind of a big challenge. But then also, even though we have like found out many genes, doing some expression analysis, testing the markers kind of helps to narrow that down. So that's my experience.
Okay, thank you, Swati.
I might jump in. Just one quick comment. So I agree with everything with Swati just mentioned. I think my work is a step behind because basically we are trying. so as I mentioned, I'm working with 3 diseases like in this presentation, and Downy mildew is the most important worldwide for lettuce. And the idea is to try to find accessions with multiple disease resistance, because in a breeding program, like it takes forever, 8-10 years to develop a resistant cultivar. So PTL analysis helps to, decrease this time. But in my perspective here, from my research, we try to use some accessions, or that is not cultivars yet, to be tested with other pathogens. And then if we can combine in one single accession multiple disease resistance, we can breed elite breeding lines to incorporate in the breeding program. So we are just trying to find as many tools as we can to improve as faster as we can the plants.
I'd like to add to what Liz said, if we have some time. Yeah, so sometimes, especially when we're working with certain pathogens, the resistance, it's likely to break down at higher temperature, and under lower temperature, it's still the species could be resistant. So sometimes when we do identify QTLs and use it for breeding purposes, but eventually at certain different temperatures, it might still not work. So it's not exactly a challenge in identifying QTL, but it is a challenge after, after applying the QTL that was identified, if that adds up.
Yeah.
Thank you.
If I could jump in just one more time. So as like the theme of this symposium is climate change, and it's believed that outbreaks of diseases, in our case that we work with diseases, are being more frequent. So doing this kind of analysis is very important, because like even though we breed for many years or shorter years a variety that is resistant, the outbreaks are happening. So the breeding is a continuous work. So yeah.
Yeah, truly so. It's definitely continuous work. It's always going. Yeah, thanks so much, guys. I think we have enough time for maybe one more question. We have a question for Kar. What are the characteristics of good flavor?
Okay, so as I mentioned in my slide, sugars, acids, and volatiles, I said 400, but our lab narrowed it down to like 30, 33 that we usually test the varieties for. So when you ask for what are the, I can count like a few only, I can't do that, but we have like a profile, a set of volatiles that are, that's why it's so complex, because of the interactions and overlapping pathways and all. So overall, you can say sugars, acids, and a bunch of volatiles in short. Thank you.
Thank you. Yeah, I've had many different tomatoes, and they seem to all have very different flavors. Sometimes you get a tomato that tastes bland like nothing, and sometimes you just get that super juicy, sweet, acidic tomato, and it's really, really good. So yeah, your work is much appreciated in making those tasty tomatoes.
Thank you so much.
All right, guys, so we're getting close up to lunch here, so I think I will go ahead and we'll wrap up the Q&A. You guys all did a very good job, great presentations, great Q&A sessions, really good questions, everybody. So we'll go ahead and we'll do lunch, and so that will be that'll go up to 12:55, at which point we will have our next speaker, and I will be handing over the reins then. In the meantime, I will be tallying up your guys' points from our 3 judges. So if those 3 judges want to get those score sheets back to me ASAP, that would be great. And then during the closing remarks and award ceremony, I'll go ahead and I will announce our 3rd, 2nd, and 1st place winners. So if we can just do one more round of applause for all our presenters today. Thank you guys so much. You guys did great.